everybody, and welcome one more time to our online lectures. In this video, we will continue with our discussion in Chapter 5, specifically the section on our nucleic acids. So let's jump right in. Let's talk a little bit about the roles of nucleic acids. We have two different types of nucleic acids. We have DNA and we have RNA. Those are the molecules that are in charge of coding for all of the genetic information in a living organism. DNA stands for deoxyribonucleic acid, and RNA, which is a derivative of DNA, it stands for ribonucleic acid. DNA provides directions for its own replication. That means that DNA or RNA, nucleic acids in general, are molecules that are self-replicating with the help of certain enzymes. The way that DNA is constructed, it provides for its own replication in a very specific way. DNA directs the synthesis of messenger RNA, which is for short mRNA, and through mRNA it controls protein synthesis. This process, it's called gene expression. So if you were wondering what kind of information can I find in a gene, we know that genes are sections of DNA that encode for a certain amount or a type of information. That information will translate into a protein through the functioning of messenger RNA. And we're going to see later on that mRNA pretty much functions like a recipe with it marks the specific ingredients that are necessary for making a protein. And applying what we know already about proteins, that is the specific sequence of the amino acids that will make up that protein. So let's have a look at this stuff. Number one, in the process of making proteins, parting from DNA, the first thing that has to happen is the synthesis of RNA. And this is called a process of transcription or rewriting DNA in another language, and that language is going to be mRNA. So in the process of gene expression, the first thing that we have to do is we have to synthesize or create a molecule of messenger RNA, and it will be in that section or in that little segment of mRNA that I'm going to have the specific order in which I am going to assemble my amino acids to finally form that protein. So after I create the mRNA, that mRNA is going to move outside of the nucleus. This is created inside the nucleus, so it will move out through a nuclear pore, and that mRNA now is going to be deposited out in the cytoplasm of the cell. Who is going to take care now of the action of the gene expression? The factory for proteins. So we have been talking about this since day number one. The ribosomes are the organelles in charge of the synthesis or production of those proteins. So a ribosome is going to read the information that it is coded in the mRNA molecule, and then that will promote the synthesis of that polypeptide following the instructions on that mRNA. Just like making a cake, just like cooking from a recipe, the ribosomes are going to follow the instructions encoded in that mRNA molecule. And as the polypeptides are added to the chain, or the amino acids are added to the chain of polypeptides, then I am getting a primary structure of a protein, and eventually a secondary structure, tertiary structure, and quaternary structure if it is needed. And this is pretty much the process of transcription and translation that eventually we'll put into much more detail. Transcription means making an RNA molecule parting from DNA, and transduction, which is kind of like changing the language, we are changing the nucleic acid or using the nucleic acid to construct protein. So that is the process of transcription and translation. Okay, so more on the roles of nucleic acids. Each gene along a DNA molecule directs the synthesis of a messenger mRNA. So the DNA has the instructions, it had the sequence that will be then transcripted into an mRNA molecule. The mRNA molecule interacts with the cell's protein synthesizing machinery to direct production of a polypeptide. So in this case, the ribosome, it's the worker that will use the mRNA and it will promote the protein synthesizing machinery, which is pretty much just producing those proteins. The flow of genetic information, this is going to be a very important one, can be summarized as DNA moves into RNA, and from there I end up getting proteins. And this is what's called the central dogma of information, in which information from genetic origins will move in one direction, from DNA to RNA, eventually into a protein. So very important to keep that in mind when trying to understand and the functioning of these molecules. DNA is the original one. RNA, it's another kind of nucleic acid that the ribosomes can understand, and the ribosomes will put the amino acids together to form that protein. And that is the flow of genetic information. So what is in nucleic acids? Nucleic acids are polymers. They are called polynucleotides. Remember that polymers are repeating subunits of very similar monomers, and in this case we're talking about nucleic acids. Their specific name would be polynucleotide. Each polynucleotide, it's made of monomers that we call nucleotides. Each nucleotide consists of three important things. One of them is going to be a nitrogenous base, a sugar, in this case specifically a pentose sugar, and one or more different phosphate groups, which will eventually make the backbone of the DNA molecule. Here in this illustration we have a representation of what a nucleotide is going to be. It has a nitrogenous base, and this is what we know as the four letters of DNA. Adenine, guanine, cytosine, thymine, and in the very special case of RNA, the letter U or uracil. Uracil is exclusive to RNA. There is no letter U in DNA. That nitrogenous base, it's attached to pretty much the same thing all the time in all of the different nucleotides that we have, any of the 5 nucleotides that we have. This is the sugar pentose and the phosphate group, and in this other more generalized picture we can see how the nitrogenous bases are the only things that are going to change in the nucleotides, while the ribose or the sugar and the phosphate group will remain constant, making that backbone of DNA. The portion of a nucleotide without the phosphate group, it's called a nucleoside. So how do we generalize this? A nucleoside, it's the repeating subunit that will make the polymer, and what makes each polymer different from one another, it's going to be that nitrogenous base, the A, the G, the C, the T, or in the case of RNA, the letter U. So the nucleoside, it's just the nitrogenous base plus the sugar. There are 2 families of nitrogenous bases, and this is going to be very important. All of those letters, I can subdivide them into categories: the pyrimidines and the purines. It's very important to kind of like study those names because they sound weird. They're a little complicated in structure. It's hard to, it's kind of like hard to remember about something doing mnemonics, so keep that in mind. The pyrimidines are cytosine, thymine, and in the specific case of RNA, it's going to be uracil. The purines are adenine and guanine. They have a 6-membered fused ring to a 5-member ring. So in the case of purines, they have like the larger kind of like structure in terms of the nitrogenous base because they have 2 fused rings, together. One little mnemonic that I do that I can use for remembering this, I take the length of the words. Pyrimidine, that is a very, very long word, so I think always pyrimidine. Too complicated. Letters T and C for thymine and cytosine. And then purine, it's a lot simpler for me to remember, less words, so I can say it's all good, and that is the A and the G, so that it's adenine and guanine. So too complicated, pyrimidine, all good, purine, and that's kind of like can help you with a little guide onto how to remember this. In DNA, the sugar is a deoxyribose, and in RNA, the sugar is a ribose. So if we're looking for, at least structurally, for a difference between RNA and DNA, it would be the sugar that it is enclosed in the nucleoside, deoxyribose in DNA and ribose in RNA, and that it's a big difference between DNA and RNA. Remember that at the end of everything, it will be very important that we know and understand the differences between RNA and DNA. So, what is a nucleotide altogether? It would be the nucleoside plus the phosphate group all together will make me a nucleotide. So, when we are thinking about what is a nucleotide, it's not just the nitrogenous base. It's the nitrogenous base that is bonded together with the other two segments. So a nucleotide would be the phosphate, the sugar, and the nitrogenous base. Very important to keep that in mind. Here we have another animation that you can observe if you download the presentation for you to study and look at something a little bit different. So feel free to watch that by yourself in your study time. Let's talk about nucleotide polymers. Nucleotides are linked together by a phosphodiester linkage. This is the name of the specific bond that holds the nucleotides together. And when this happens, they build a polynucleotide. So just like the other polymers that we have been observing up to this point, they always have a covalent bond that puts them together. So I would recommend, it's a good idea, write down all of the names of the molecules that we are discussing, specifically the three of them that make polymers, and put to their side the name of the specific bond that creates them or puts them together. The one for the carbohydrates, for the amino acids, and for the nucleotide polymer. So in this case, it's a phosphodiester bond, and this is the bond that holds together. Right, this little bond right here, it's the one that puts together the bases. These links create a backbone of sugar phosphates, and in combination with their nitrogenous bases, they form the DNA molecule. The sequence of bases along the DNA or the mRNA polymer, it's unique for each gene. So this is kind of like something to talk about, very important as well. What is a gene? So now we can see the gene in a molecular kind of way, because it's not only we're thinking about genes as little segments of DNA with specific information. Now we know where the information is coming from. It's coming from that sequence of nucleotides that make that gene. So all of the genes are going to be different from one another because the nucleotides that are inside of that gene are actually different from one another. So I can make different mRNAs based on this gene, and then the ribosomes are going to make specific proteins based on those specific letters. How amazing this little thing is. It's really, really, really mind-blowing the way that our cells, our nuclei, and our DNA actually codes for things. There's a very, very specific way, really easy to understand as well, because it's just like a recipe. All right, moving on. Talking about the structure of DNA and RNA molecules, DNA molecules have two polynucleotides, sorry about that, spiraling around an imaginary axis, forming a double helix. So what happens is that these two molecules are wrapping around one on the other, forming some sort of staircase, some sort of spiral staircase, moving up or down, depending on how do you want to see this. DNA, it's a double-stranded membrane. One membrane, it's complementary to the other strand. Okay, we'll get more to that. We'll get more on that in just a few minutes. The backbone of DNA runs in what we call an opposite direction, from a 5 to 3 carbon. We'll get to that. Let's not think anything about these numbers yet. Let's think about the 5 and the 3 as a direction, like a road, like a one-way road. And the name of that road is the 5 to 3 direction. Okay? They run anti-parallel one with the other. That means, just like a road, one one lane goes in one direction and the second lane runs in the opposite direction. So if we think about the strands of DNA, one of those strands is going to run in the 5 to 3 direction, and the other one is going to run in the 3 to 5 direction. So let's look at this picture that we have here that illustrates this particular concept. Here we have two strands of DNA. One is going in the 5 to the 3 direction, and the other one, the complementary strand, it's running in the 3 to 5 directions. And why is this occurring? Why are these two strands joining together? It's because bases, or the bases of DNA, they make pairs with their complementary base. Who makes the pairs? Well, A pairs with T and C pairs with G, and because they're really complementary to one another, they get close together. They're close together by hydrogen bonding that we talked about, that weak attraction between one and the other, and that's pretty much what makes a DNA molecule being a double helix molecule. Needless to say, one DNA molecule includes many, many, many genes. So in the case of humans, we have 46 chromosomes in our DNA, and each one of those chromosomes contains thousands or tens of thousands of genes in that structure alone. Only certain bases in DNA pair up to form hydrogen bonds, so it's not a free-for-all. The bases can't randomly attach to one another. They are like a Romeo and Juliet of molecules. They have this soulmate that they will only pair with. So adenine, or the letter A, would always pair with the letter T, and guanine and cytosines will pair as well. That means that you are not going to find A's pairing with A's or with G's or with C's. They would only pair with T's. This is called complementary base pairing. This feature of DNA structure makes it possible to generate two identical copies of each DNA molecule in a cell that it is preparing to divide. And we have talked about this replication of DNA both in the cell cycle when we discussed the S phase of interphase, and also when we talked about mitosis and meiosis. There is always this replicating stage in DNA, and this is what happens because only one pair would pair with another pair. This is going to be an exclusive thing, one from the other. If I separate these strands, I can use the strand to make its complementary strand, and that way I can create identical copies of this DNA molecule. Very needed for DNA, very needed for the cell cycle. Now, RNA, in contrast to DNA, it's a single-stranded molecule. RNA, I'm not going to find RNA as a double-stranded molecule, and there's a reason for that. It's because ribosomes have to read the letters, and if the letters are complementary to one another, they are hidden away from the ribosomes, and I can't read them. Complementary pairing can also occur between two RNA molecules or between parts of the same molecule. Sometimes this happens as well. Like we can see in the picture here, for example, sometimes the molecule folds into itself and it finds a complementary section that the molecule can bind to. And it creates this weird shape of RNA. So RNA is not always linear, right? It's not always completely linear. In RNA, this is very important, very, very important. In RNA, thymine is replaced by the base uracil. So the letter A will only pair with the letter U. And this will only happen in RNA. Let's remember, because in RNA, there is no letter T. So if I show you a sequence of nucleotides, and you see the letter T in those nucleotides, you can immediately assume that I am talking about DNA. On the other hand, if I show you a sequence of letters of nucleic acids or nucleotides, and you see a letter U in there, then that would indicate that this is an RNA molecule. If you see both if you see both T's and U's, this would be this mistakey molecule, so be paying close attention to that. They can't be T's and U's in the same molecule. Kind of like a very interesting thing. Here we have another animation that you can find later on the presentation if you would like to do so. Once the structure of DNA and its relationship to amino acid sequence was understood, and this took a long, long time, biologists thought to decode genes by learning their base sequences. So we could understand proteins a lot better because we could code, we can find the order of amino acids, and we can link those to specific letters on RNA, and from there we could understand where on a DNA molecule a specific gene is by sequencing these letters. Isn't that amazing? It's absolutely amazing. The first chemical techniques for DNA sequences were developed in the 1970s and have been refined. They have been refined over the next 20 years. So our understanding of the molecular functioning of DNA has been a lot better. This has led us to understand proteins, understand disease, understand mutation, understand how evolution is working by simply observing the sequences of the nucleotides that make up the DNA molecule. It is enlightening to sequence the full complement of DNA in an organism's genome. This is like reading all of the instructions of every single little thing that you can find in an organism. The rapid development of faster and less expensive methods of sequencing was a side effect of the Human Genome Project, which was an enormous project to sequence all of the letters in the DNA of humans. This was an enormous thing, an enormous achievement of science in the last century. Many genomes have been sequenced, generating large sets of data, which are the fundamentals of our genetic understanding of life. And it all has to do with nucleic acids, with DNA, with its derivative RNA, and how this puts or gets together to form the proteins that allow the functionings of living things. So that concludes the chapter 5 on biochemical molecules. Hope this was enlightening for all of you, that we have a better understanding of those 4 very important molecules that make life what it is today and make life as we know it. So thank you for your attention, and I will see you on the next one.